Hey everybody, we're in Chapter 8, which is all about genetic engineering and biotechnology. This is a very fun chapter, in my opinion. Before we talk about genetic engineering, let's look at how bacteria can genetically alter their genes. And so we have two different mechanisms that can cause alteration to genes: vertical gene transfer and horizontal gene transfer. So vertical gene transfer occurs when a parent passes their genes on to their offspring. And in that process, if a parent has some type of mutation, then the offspring can also get that mutation, changing their genetics. Horizontal gene transfer occurs when we have two cells in the same generation. One cell offers another cell genetic information. So there are three ways that we can horizontally transfer genes. One of the ways is called transformation. This is definition-wise taking in naked DNA from a solution. The second one is transduction. Transduction is transferring DNA from one cell to another cell via a bacterial virus or a bacteriophage. And then conjugation is transfer of genetic information from one cell to another cell through a sex pelus. So transformation was discovered by Frederick Griffith, and he discovered this back in 1928 before we really knew that DNA was what was being transferred from parent to offspring. And so how he discovered it, he was working with two strains of streptococcal bacteria, a smooth strain which was encapsulated, and then a rough strain that was not encapsulated. And what he noticed was when he would infect rats with the smooth streptococcal bacteria, the rats would get a streptococcal infection and they would die. And when he looked at their blood, he noticed that there were smooth streptococcal bacteria that he could isolate from the rat. Then he also did this with the rough strain of streptococcal bacteria. And the rats that had the rough strain did not die from from an infection. But when he killed those rats and looked at the cells within, and then plated the blood, he found again some cells. They were the smooth streptococcal bacteria. So at this point, he's thinking, okay, maybe it's the the capsule that is causing the pathogenesis. Maybe the bacteria don't cause anything because the rough bacteria that didn't have a capsule didn't kill the cell and they were still there, so there's something wrong, right? So he killed off the bacteria that were in the capsules, so it was smooth bacteria, but they were dead, and he infected rats. And this time he found nothing. He wasn't able to isolate anything. So then he's like, okay, so smooth killed and rough unkilled don't cause infections. Let's put them both in and see what happens. the rat actually got sick and died. And when he looked at what type of colonies, there were both smooth and rough colonies found. And he was totally baffled. He was like, what? What is going on, you know? And what he determined was that the smooth killed bacteria, the DNA from them was leaking out into the extracellular region. The rough bacteria that were in the solution with the killed bacteria took in some of that DNA through transformation and were able to then form a capsule. And the encapsulated living smooth cells killed the rat. So for cells to be able to take in DNA via transformation, they have to be competent. So competence isn't, when you think of competence, you think of, knowledge, you understand something, you're competent in that subject. But when I'm saying this, what I mean is they have to be able to take in DNA. So if you're a competent cell, you can take in DNA, kind of like if you are competent in a subject, you take in that subject and you have that knowledge. Okay? So if you're not competent, then you cannot take in the DNA, and that means you're not going to be able to be transformed. Conjugation is the only mechanism of gene transfer where bacterial cells have to touch. And so with conjugation, we have two different types of bacteria. You have a plus cell and a minus cell. The F factor is a factor found in plasmids that allows for bacteria to produce a conjugation pilus or sex pilus. And once it does, that pilus will move from one cell to another cell, and that's where the genes will be transferred across. So typically through through conjugation, we're transferring a plasmid that contains the F factor plus other information. And so when you have a negative and a positive cell, when the negative cell takes in that plasmid, it becomes a positive cell because now it has its own F factor. And so here's what it looks like. Here we have bacterial chromosome. Here we have the plasmid. The plasmid is going to which codes for that sex pilus, produces the sex pilus. So it transcribes and translates that sex pilus and produces it. The plasmid then undergoes replication, and one of the plasmids moves across the sex pilus into the F minus cell. And at the end, we have two F plus cells. Transduction then is the movement of genes from one cell to another using a bacterial virus or bacteriophage. There are two types of transduction, generalized and specialized. In generalized transduction, a bacteriophage infects a bacterial cell. And when it does so, it causes the bacterial DNA to get broken up, and it produces its own genetic information. New viral structures assemble within the cell, and they take in the genetic information. Most of them take in genetic information for the virus, but sometimes you have a viral structure that takes in genetic information associated with that bacterial cell. And so when that bacteriophage infects another cell, which is what you see here, it releases the DNA from this bacterial cell here into this new cell, and that DNA then incorporates itself into the genomic information of the cell and becomes part of that cell's genetic information. In specialized transduction, we are choosing the information that we care about. And so we produce a bacteriophage that we infect certain bacteria with. The bacteriophage releases its genetic information into the cell. The information that enters the new cell can incorporate itself into the genetic information of the bacteria, and then it can lie dormant within that cell. So that's a lysogenic cycle. And then when needed, so we now have new information that we care about in the cell, that cell can replicate multiple times, and when needed, we can excise that information along with neighboring information from the actual bacterial cell that we care about. And so then that bacteriophage enters the lytic cycle, pulls the information out after producing new viral particles, and takes that new information and can move it to another cell. That's specialized transduction. I did have a video that I'll show you.
Specialized transduction involves the transfer of only a few specific genes from one bacterial cell to another by means of a phage. The lambda phage, which infects E. coli, is a well-studied example of a specialized transducing phage. When lambda phage infects E. coli, the phage DNA enters the cell and then integrates. Specialized transduction involves the transfer of only a few specific genes from one bacterial cell to another. Phage particles are produced. The lambda phage, which infects E. coli, is a well-studied example of a specialized transducing phage. When lambda phage infects E. coli, the phage DNA enters the cell and then integrates into a specific site on the host chromosome. When an E. coli culture carrying the lambda phage is induced, phage particles are produced. On rare occasions, a piece of bacterial DNA, for example the GAL gene, near the specific site of insertion remains attached to the phage DNA, and a piece of phage DNA is left behind. The phage that develop are defective because they do not carry the entire phage genome, but can still infect other cells. The defective phage can attach to another bacterial cell, and the DNA can be injected. Both phage and bacterial DNA
Wrong
will
one.
become
That's where we're
integrated into the new host.
So we use these different mechanisms of horizontal gene transfer to alter microorganisms. And that is genetic engineering, altering microorganisms or altering any organism's genes to allow them to produce new useful substances or to alter their characteristics. So we use the term biotechnology is the use of microbiological and biochemical techniques to solve certain problems, such as being able to identify who the baby daddy is, or being able to identify who the killer is from a list of potential suspects, being able to identify the potential for a certain disease that runs in your family. Those are reasons we use biotechnology, producing organisms that have characteristics like bacteria that can clean up oil spills. I feel like I missed it. Okay, no. Okay, yeah. Okay. So to utilize or to alter genetic information, we have to use certain tools. And one of those tools is called restriction enzyme. Restriction enzymes are proteins produced by bacteria. They're named for the bacteria that produce them. And what they do is they cut DNA at a specific sequence. The sequence is typically palindromic. And so a palindrome is something that is read forward and back the same way. So here you see GAATTC, and down here you see GAATTC. So this is a palindromic sequence. Restriction enzymes can produce fragments that are blunt or fragments that have sticky ends or cohesive ends. We typically use restriction enzymes that produce sticky ends. What sticky ends allow is for us to take and cut DNA from one a section of DNA or chromosome that contains a specific gene that we care about, and then we cut another piece of DNA that we care about, and we hybridize the two together. We allow them to come together, and the complementary bases will attach. Once we do that, we then use ligase to covalently seal the bonds between the sugar phosphate backbone. We named restriction enzymes by the bacteria that produce them. So many bacteria produce restriction enzymes. Restriction enzymes actually are the immune system that bacterial cells have. So they produce these restriction enzymes to fight off the one infective agent that causes problems to bacteria, and that is the bacteriophage or the virus. So phage DNA and bacterial DNA are composed of the same nucleotides, A, T, G, and C. So if we can use an enzyme that cleaves DNA at specific regions, so like this ALU1, Arthrobacter luteus, produces a blunt cut right here between AG and the CT. This will cut up the viral DNA, and the viral DNA is no longer able to produce its proteins properly. So that's what the restriction enzymes function for. But we use the sticky end ones, so like this Bacillus amyloliquefaciens H is known as BAM H1. And it produces a sticky end between, so the letters are GGATCC, GGATCC. Between the two G's here and the two G's here, we produce the sticky end. And then we can cut DNA that we care about that contains maybe the genes for insulin. We hybridize them together and infect new cells with them. And when we do that, we produce bacteria that can that can produce now the insulin that we have just added to their genome. So this has allowed us to produce human insulin. Back in the 60s and 70s, even in the 80s, people were using bovine insulin. This allowed bacteria to, or this allowed people, like they would get pig insulin or other types of insulin, but not human insulin. And so if you had diabetes, type 1, juvenile diabetes, you were using another animal's insulin in your body to help you break down glucose. Actually, that's not true. To help you take in glucose into your cells so that it wasn't in your blood. What this did was it allowed us to produce human insulin in mass amounts so that we could use that to be able to supply patients that had type 1 diabetes with the medicine that is effective for their bodies without costing a lot of money. We also use biotechnology to produce bacteria that are able to break down certain chemicals like oil for oil spills, or to produce other antibiotics, or other important drugs like human growth hormone. In all of these, you have to have DNA cloning to allow us to produce enough of whatever chemical we care about so that it can be utilized in the environment. So how does this work? We're going to take DNA that we care about, isolate the gene that we care about, cut the DNA with a restriction enzyme so that we can take out that gene that we care about. We then cut a plasmid with the same restriction enzyme, and we allow the DNA and the plasmid, which is the vector, to hybridize together in a solution. Once that happens, we add E. coli, and then E. coli, which typically is not competent, but we can shock it to make it competent. What shocking does is it breaks open the cell wall so that the DNA can be taken in. And then the competent E. coli will take in the new information and we'll be able to utilize the information. And so there's that electroporation where we're shocking the cell to break it up so that the bacterial cell can take in the information. If we use a high copy vector, the vector can replicate multiple times within the same or within an individual bacterial cell, and then when the bacterial cell replicates, multiple copies will move into each cell, and that allows us to produce millions of copies of the same gene even faster. And so we can produce like lots of insulin, which then can be used to, or can be harvested to give to patients who have type 1 diabetes. So how do we actually do this? We have to have a vector that has certain characteristics. It has to have an origin of replication, where it's able to replicate on its own. It has to have 2 different selectable markers. So the first selectable marker is usually a resistance factor that allows it to survive in an environment with certain chemicals, so like an antibiotic resistance. A very common selectable marker is ampicillin resistance. A second marker helps us to distinguish between cells that took in the plasmid from cells that took in the plasmid with the genetic information we care about. So we'll always have some cells that just take in a plasmid, but no genetic information, and other cells that took in a plasmid that also the plasmid has in it genetic information that we care about. And once we have this nice vector, we then can infect cells with the vector, and we can then plate the bacteria on a medium containing ampicillin and whatever gene that may have been disrupted. So, containing the ampicillin resistance, as well as the chemical that we have disrupted if we have the proper gene. And so in this case, we use a vector that has a LAC-Z gene. The LAC-Z gene allows us to produce a protein that breaks down lactose. So if the LAC-Z gene is intact, it will break down lactose, producing blue colonies. If the LAC-Z gene is not intact, then we don't have that functional protein being produced, and so we won't utilize the lactose and we'll produce these white colonies. And so this is known as the blue and white screening test. And it helps us to identify the bacteria that have taken in the genes that we care about. And I think it's right here. So let's look at this a little bit closer. When we're cutting a plasmid and we're cutting our DNA that we care about with the same restriction enzymes, and we put them in a solution, we can't see them. This is microscopic. So you think that the bacteria that you look at under a microscope are small. This is uber small. These are macromolecules that we're talking about. And some are even smaller than that. They're like peptides. So not even a full protein or anything in size. So we don't really know if we cut the plasmids all properly, if the DNA was cut properly. We're not going to know if the plasmid takes in the DNA that we care about or doesn't take in the DNA that we care about and just hybridizes together on its own. Then we add bacteria, typically E. coli. E. coli are some of our favorites. We add the bacteria, and again, we don't know which bacteria have become competent. We shock them to make sure that they have the ability to be competent, but sometimes some bacteria take in the plasmids and some bacteria won't. If they do take in a plasmid, they'll have resistance to ampicillin, but they may not take in a plasmid that contains the gene of interest that we care about. And so with that, hold on a second. And so when we actually plate the bacteria and let them grow, we're going to have E. coli growing and making colonies. So we have to use more than just a nutrient agar. If we use a nutrient agar that we've added ampicillin to, now we're going to have less colonies grow because only those colonies that contain the plasmid will grow. But we can even narrow it down further so we can find the bacteria that contain the plasmid and the gene of interest by plating it on a medium containing both the ampicillin and lactose. So if the cells utilize the lactose, that means the LAC-Z gene is still intact. Whereas if they do not utilize the lactose, that means something was inserted within that gene and those are the white colonies. And so we know we can pick these white colonies off the plate and grow just the bacteria that we care about that contain our insulin. And so I use insulin as my example, but insulin is just one example. We can also use plasmids and bacteria to produce plants that are resistant to herbicides, or to produce growth hormone for people that are not producing enough growth hormone. We also use DNA or biotechnology to help us identify mutations within our own genome, specifically so we can know if a disease is going to be more common or less common. Or I think and is the better one. We can use this to identify the suspect at a crime scene as the actual, person who caused the crime, the criminal. So in this case, we're going to have to get DNA from something. If it's at a crime scene, we might be picking up small amounts of blood or tissue from under their fingers. If we're working with a patient, we might just take some blood so that we can then run tests on it. So I showed you, or we went through how cells can produce genes that we care about. Sometimes, well, no, not sometimes, but doing that takes time. I mean, you first have to grow the cells, and then you have to pick out the cells that you care about and then grow them only. And so it's a more time-consuming process. It's not hard, but it's time-consuming. And we have come up with new processes that don't use cells at all. And so this is known as in vitro replication of DNA. And the process is known as polymerase chain reaction. This should say amplify, AMP. I just noticed that. We're not using cells. This entire process was thought up by this man here. This is Dr. Kerry Mullis. He is a scientist. He's also a surfer. And when he was out one day catching the waves, doing his cool, rider stuff, he was, possibly smoking some dope. He was, kind of a free-spirited man. And this occurred in, the 80s, and so this stuff happens. He was out there thinking, and he was like, I wonder why we can't just grow these cells inside of, or this DNA inside of test tubes. If we take all the proper things, the DNA, the nucleotides, we had primers, we had buffers to make sure everything stays, works effectively. We add our DNA polymerase, and then we just heat the cells up to proper temperatures so that we can have the DNA come apart. We can have primers anneal. We can have DNA polymerase come along and digest the primers and produce new strands of DNA that we care about. And so he went back to his office and he started fiddling with this and came up with this process, which we use today still. It's a little bit more high-tech than what we did before. So this is a PCR machine from the 80s, very high-tech here. You have 3 water baths. Each water bath is going to be heated to a different temperature, somewhere around 92 degrees Celsius, 54 degrees Celsius, and 72 degrees Celsius. And we're going to move small test tubes from this water bath to this water bath to this water bath, and then we're done. And then we grab more test tubes, this water bath, this water bath, this water bath. And we just keep doing it until we are done. So what this does is it amplifies DNA very rapidly. And we can use the genes, run the genes on a gel to identify DNA fingerprints or to identify specific mutation. So the 3 steps of the polymerase chain reaction are denaturation, which allows us to take the DNA apart. We then have the anneal step where where an RNA primer is allowed to anneal to a specific spot that we care about. So we produce the primers so that we're going to only produce DNA that we care about. And then we raise the temperature back up to 70 degrees Celsius. We have to add in our DNA polymerase, and then we allow the DNA polymerase to extend. So the DNA polymerase we use today is called Thermus aquaticus. It's called Taq polymerase, and it's produced from Thermus aquaticus, which is a heat-stable bacterial species. This allows us to add DNA polymerase once, and since enzymes don't get used up, it can be reused over and over again. What we end up receiving is DNA. So we produce multiple strands of DNA. We have the long strand, we have medium-sized strands, and then we have the majority are short strands of DNA. And these short strands are what contain only the gene of interest. So they are the ones that we care about. So I do have a video I'm going to show you, but first we're going to come over here and I'm just going to show you what happens. So here we have multiple cycles before we even started amplifying the DNA. We have 2 strands of DNA. At the end of each cycle, we're going to double the amount of DNA. So if we start with 2, we produce 4, and then we have 8, and then 16, and then 32, and then 64. And so how does this work? Every long strand produces a medium-sized strand. So wherever that primer anneals to, it's going to allow us to produce a medium-sized strand of DNA that contains the genes that we care about plus a little bit more. At the end of the second cycle, the long strands still produce medium-sized strands, but the medium-sized strands produce these tiny strands that only contain the gene that we care about. At the end of that cycle, we now have long, medium, and small. So the long strands still produce more medium strands, the medium strands produce more small strands, and we have small strands now that produce more small strands. So within just a few cycles, we can have thousands of copies of the DNA, millions of copies by the end of the 30th cycle. And this is the same song, so. Amplified DNA. I had to grow tons and tons of tiny cells. And along came a guy named Dr. K. Said you can amplify vitro just as well. Just mix your tablet with a buffer and Nucleotides and polymerases do. Denaturing, annealing, and extending. Well, it's amazing what heating and cooling and heating can do. PCR when you need to detect mutation. PCR when you need to recombine. PCR when you need to find out who the daddy is. Who's your daddy? PCR when you need to solve a crime. PCR when you need to detect mutations. PCR when you need to recombine. PCR when you need to find out who the daddy is. Who's your daddy? PCR when you need to solve a crime.
To all the scientists out there doing PCR, Biorad salutes you with the all-new 1000 series thermal cycling platform.
So I told you that the mechanism that we use has become a little bit more elaborate. Back in the 80s, we used the three water baths, and we would have people that had bachelor's degrees that would stand there for 8 hours a day. I mean, they'd get breaks, obviously, but that was what their job was. They would be moving the DNA from one bath to another bath to another bath, and then they'd add more DNA polymerase and then go back again through it and just constantly do the same thing over and over and over again. But once we found the Thermus aquaticus and were able to use the DNA there, we no longer had to re-add DNA polymerase after the denature step. And eventually we were able to produce a machine that would heat up again multiple times, and that's that thermocycler. So now, instead of having scientists do that, we put everything into a tiny machine, push a button, and it will go through 30 cycles and we can basically just go about our business. We don't have to do anything else. And most of them will even go into a freezing stage at the end so that you don't have to worry about being there when the DNA or when the replications end so that you don't ruin the DNA. So it's awesome. And that song was awesome. I love it. If you remember the 80s at all, so you'd have to probably be in your 30s to remember the 80s, but you may remember the song anyways, because I mean 80s music and the 80s were the bomb diggity, right? We had a period where there was a great famine in Africa, and we were trying to raise money to provide food for the people in Africa, and a bunch of singers got together and produced a song that was called We Are the World. So this PCR song was a play on the song We Are the World, and it was awesome. Anyways, I love it. So on your exam, you'll definitely have to do at least one PCR cycle. And so this is how you would do it, or what you might be given. I might give you a number of strands of DNA. And what you have to know is that at the end of each cycle, the total number is going to double. So, hold on. So what you want to remember is that whatever the long strands are that I give you, that number is always going to stay the same. And at the end of each cycle, you're going to have a doubled amount of DNA. So at cycle 0, that means we haven't started. We only have 5 strands of DNA because that's what I've given you. Each long strand always produces a medium strand. Each medium strand always produces a small strand, and each small strand produces a small strand. So at the end of the first cycle, we will have 5 medium strands. These 5 strands will never leave, so they just move over. At the end, so you add these up, that gives you 10. If you do it again, now you have, again, 5 more medium strands produced. And these 5 medium strands produce 5 small strands. These 5 strands also have to move over, and these, of course, move over. So now you have 5 plus 10 plus 5 is 20. Do it again. These move over. They produce 5 more. So you have 10 plus 5 is 15. You have these 10 medium strands that produce these small strands. Plus you have these 5 small strands that produce small strands. And these 5 small strands have to move over. So you're going to get 10 plus 5 plus 5 is 20. So 5, 15, and 20 equals 30. Do it again, and there's the last one. And you can go on and on. So we get all this DNA, and it's the small section that becomes the most common, if you noticed here. These always stay the same. These only go up by whatever number the long strands are. And then we have these small strands that become really common. We then can add a probe to the DNA. Oftentimes we put something into a gel that allows the DNA to glow under UV radiation. And what this allows us to do then when we produce or when we put the DNA into our electrophoretic gel and we run it through electrophoresis, we produce small little segments depending on the size of the DNA. So the DNA runs from these tiny wells, which are at the top, down, and the smaller segments move faster. And then where we have a large amount of DNA that settles out, that's going to tell us or give us some information about that DNA, like the size of the gene that's there. And so here's an example. Of course, examples always are perfect. I'll just let you know now. Whereas in real life, gels are not as easy to read because sometimes there's a little bit of overlap between 2 different segments. I mean, I've given you a 5, 4, 3, 2, and 100 segments. Some of these will be the same length of DNA or the same length of gel, and they might have up to 1,000 segments. So the first one is always going to be a size marker that helps you identify the size of the DNA. The second one and on is going to be whatever you care, whatever information you put in. So in this case, I put a victim's DNA, a criminal's DNA, and then 2 suspects' DNA. So once you've run the gel, then you're going to have results such as this. Of course, this is perfect, like I said, because I made it. And so here the victim's DNA settled out at 200 and 300 base pairs. Here's the criminal's DNA, and then here are 2 suspects. And so just by looking at it, you know that number 5 is the suspect that has the same DNA sequence as the killer or the criminal, whatever the criminal is, and suspect 4 doesn't. What that allows you to do is it tells you that the suspect 4 is going to be excluded and suspect 5 is going to be included as a potential criminal. And then you're going to have to run him through the law, go to court and all that stuff, and because you're always innocent until proven guilty, which, is the way it is. So we can do this also looking at genes. So the genes in a patient. So like, think about if you have a disease that is common in your family and you want to know what's the chance that I might have this disease, or what's the chance that I can pass this disease on to my offspring. In this case, 2 might code for a non-mutated gene, whereas 3 might code for a mutated gene. And in that case, then you look at patient 4 has a normal and a mutated gene. Patient 5 has only the mutated gene. So it would depend on if the mutation is dominant or recessive in this case. If the mutation is dominant, then both patient 4 and 5 are going to show the disease. Whereas if the disease is recessive, then patient 4 is a carrier and can pass on the mutated gene to his offspring, his offspring or her offspring. But patient 5 has the disease and also will pass it on. Oh, hello. So this is kind of a fun little thing that you can go through. I got this Who's Your Daddy from this website, so you can click on it, and it gives you some scenarios, and your job is to figure out who is the father of Mary's child. She knows that it's either Bob or Larry, but she doesn't know who. So she has a DNA test done, and she has to show her child's DNA, because her child's DNA is going to correlate with either Bob or Larry. Of course, it will also correlate to her. So they take the the same DNA stretch from everybody, from Mary, Bob, Larry, and baby, and they cut it with a specific restriction enzyme, EcoR1. Mary's DNA doesn't cut because she doesn't have this GAATTC sequence. Bob's does cut once, and Larry's cuts twice. So this is what their DNA looks like. Here's Mary's, very big because it never ever cut, so her DNA is going to move really slow through that gel. Bob's produces 2 strands because his was cut once, so he has 2 strands of DNA, a very long and a very short, and then Larry's cut twice, so he has 2 shorts and a kind of long, okay? So here we have all of the DNA, and of course, the baby has Mary's DNA information, but the same baby also has Larry's information. So Bob is no longer even included as the child's father. Larry's the father and is going to either be part of the life or pay child support or something. So that's the end of this chapter. And hopefully you've enjoyed it as much as I do. This is super interesting material. You guys have a great day, and I will talk to you later. Bye.